Ki te tangeta manu e karanga nei, tui i runga, tui i raro, tui i roto, tui i waho, tui e here tangata, ti he Māori ora. E na mana e na rio aura rakatira mā tēnā koutou katoa. Tēnei ka tū raro e te maru o nā mana whenua, ka te māmoe, waitaha, kaitahu, nau mai, hara mai, tauti mai. Ki te whare tūnei, tēnā koe, tū tonu, ina mate haere hoki atu mā ki te pō. Remembering those that have gone before us, I would like to take this time to acknowledge loss of a former member of the university community, Bart Hallandorn. Bart's wife, Hupia, is still an active member of the Dunedin creative community, does work on verbatim theatre, but for a long time in her career supported us as a general staff member in the School of Surveying. Ko Waiau, ko Richard Blakey Takawingawa, he kei mahi aha te wharewananga o tākou, tēnā koutou katoa. Kia ora everyone, I'm Richard Blakey, DVC Research and Enterprise here at the University of Otago. It's my great pleasure to welcome you to this event, which is combined lectures for the Distinguished Research Medal and the Carl Smith Medal, for which we have the pleasure in front of us of 3 lectures. From Professor Keith Gordon, Professor Ben Chantal, and Associate Professor John Shaver, the latter two as the co-recipients of the Carl Smith Medal. We have the recipients, PVCs, or an alternate for the PVC in one case, to give formal introductions to the lectures. But as I chair the panel that advises the Vice-Chancellor on the award of these medals annually, I'll first briefly introduce the awards to you, their nature and significance. So firstly, the Distinguished Research Medal, which is our highest individual research award at the university, which is governed by regulations that state that, quote, the basis for selection will be outstanding scholarly achievement, including the discovery and dissemination of new knowledge, the development of innovative technology, and or the development of concepts that lead to significant advances in activities of the university. It's awarded to work that has been done primarily at the University of Otago, and it covers all of the academic pursuits across the university. Every year, all of the 4 academic divisions are invited to receive recommendations from their departments for the best and brightest researchers, research teams, and to get references from international referees to support the nominations. So it is a very, very broad basis that we have the committee deciding these awards. They're fascinating discussions, but unlike other awards like the Nobel Prizes, for example, for which there are up to 3 awards that can be made every year in each of the disciplines, we have the unenviable position of just making one award across all areas, and we have to balance overall career-long scholarly achievement with discrete world-changing discoveries and try and balance disciplines across each other. Now, just putting the scale of the exercise into context, we have approximately 1,600 academic and research staff at the university, with only 1 Distinguished Research Medal awarded each year since 2004. So there are currently a dozen and a half members of the club of recipients of the DRM. So that's only 1% of our academy will ever hope to be into this territory. Also, that then is an academy that is highly selective for entry. Very often we get more than 100 applicants for each position we have into our faculty, so there's another 1% factor. Even to be eligible to apply for an academic position usually requires a PhD or another advanced degree, which is about only 1% of the general population get to that kind of degree. So here we are recognising the 1% of the 1% of the 1% with Keith today. So congratulations, Keith, on reaching that status that I think I could say officially or semi-officially that you are the smartest person in New Zealand's smartest city today. Whether that carries on, we'll have to see. The other awards that we're recognising today are the Carl Smith Medal. That is also similarly highly valued and involves competition across all of our academic disciplines. The medal comes with an associated Roe-Heath Trust Prize as a cash award that goes along the side, the medal. And this award was established in 2003 through a generous donation from the Roe-Heath Trust to, quote, promote research at the University of Otago and give recognition to outstanding research performance of early career staff. And there are things that define early career staff of being within 10 years of PhD or career appointment. But I don't want to leave you with the impression that an early career award is any less difficult to achieve and the level of competition is any lower than for awards like the Distinguished Research Medal. I think we're doing this for the first time of putting the DRM medal alongside the Carl Smith medals, and I think you will see that the levels of achievement and the scholarly effort and the significance of the work is largely indistinguishable, albeit covering some very different disciplines. So why is it called a Carl Smith Medal? Who was Carl Smith? Well, Carl Smith was a prominent Dunedin businessman. He was the managing director of Cadbury Fry Hudson for many years and also a member of the University Council. Sir Carl and his wife, Lady Catherine, established the Roe-Heath Trust in 1964 to support the work of the university, the trust being named after the family estate of Roe-Heath in Birmingham, where they came from to Dunedin, the link to Birmingham being the Cadbury home. So I think it's quite apt and appropriate for the history of this city to have a Carl Smith Medal and a Roe-Heath Trust Award. Actually, Carl Smith was also a well-known author, in addition to his business interests, not so much for his book Sweet Success, his history of the chocolate maker Cadbury Fry Hudson, published in 1968, but also for his quirky and humorous tome called New Zealand from N to Z, that has become somewhat of a collector's piece for its interesting insights into mid-20th century Aotearoa. The book is long out of print, but what we do as a special thing for our Roe-Heath Trust Award recipients, and we may have them here somewhere, is we do dig out from the Regent Book Sale or Trade Me or somewhere copies of that book, and we may be able to give you each one of those as part of the awards that we'll make at the end of the lectures. So ati ake nei, that's a little bit about the awards, and welcome to those that are online listening to this as well. Let's get on with the show, and so I'd now like to invite Professor Richard Barker to come forward and provide the formal introduction for Professor Keith Gordon's Distinguished Research Medal. Nō reira, tēnā koutou, tēnā koutou, tēnā koutou katoa.
Deputy Vice-Chancellors, tēnā koutou. Professor Sweetman, tēnā koe. Professor Gordon, tēnā koe. Fellow recipients, Ben and John, kia ora, awesome work. Friends and colleagues, including those out in the virtual world, tēnā koutou, tēnā koutou, tēnā koutou katoa. It is my pleasure to introduce our first speaker today, Professor Keith Gordon, to give this University of Otago Distinguished Research Medal lecture. Keith graduated with a Bachelor of Science with honours in 1986 and followed this with a PhD in 1989 in chemistry from Queen's University, Belfast, UK. His PhD research was under the direction of Professor John McGarvey and focused on laser spectroscopy of solar energy compounds. He followed his PhD with a Director's Fellowship at the Los Alamos National Laboratories in the United States, where he worked on ultrafast laser spectroscopy of biological systems and solar energy materials. In 1993, we're delighted that Keith took up his post as a lecturer in the chemistry department at the University of Otago, where he remains today. He became professor in 2009, and he is currently the head of department, for which I'm very grateful. Keith was president of the New Zealand Institute of Chemistry in 2006, and he is a founding principal investigator in the MacDiarmid Institute for Advanced Materials and Nanotechnology, and he's also a principal investigator in the Dodwell Centre for Photonic and Quantum Technologies. Keith is a Fellow of the Royal Society of New Zealand, the Royal Society of Chemistry, and the New Zealand Institute of Chemistry. His research interests focus on the understanding of the properties of conducting polymers, nanostructured electromaterials such as found in organic solar cells, dairy products and pharmaceuticals, all done using spectroscopy, and he is also interested in computational chemistry. It is now time to hear from Keith himself, who is going to tell us about using light to unpick complex materials from solar cells to krill oil. Please join me in welcoming Professor Keith Gordon.
Right, hopefully you can all hear me. So thank you very much for those kind words. And yep, we're gonna just almost ready to go. Lovely, thanks a lot. So thank you everyone. So I'm going to talk today, I'm just going to put the laser on, I hope, yep. Oh, maybe not, okay. Never a good start to talk. So I've got about 25 minutes to try and explain 35 years of research, so it's going to be reasonably quick and hopefully not totally incoherent, but I do have a few things I want to try and get across. So as Richard very kindly said, I'm going to talk about solar cells and krill oil, but I thought it's kind of useful to sometimes explain where you come from, and I was going to pull out some more photos, but this seemed like enough. This is where I was schooled. This is my primary school in Belfast. I grew up in Belfast and stayed there until I was about 25. And this is my primary school, and then my grammar school that I went to, which is called Belfast Inns. This is Listonshire Primary School. Both really good educational establishments, and then Queen's University in Belfast was also a great place to do both my BSc and PhD. And someone asked me earlier why I became interested in solar energy. So it's actually to do, I realized that I kind of misinterpreted, the oil shocks occurred when I was about 10 years old, 10 to about 14 or 15 years old. But it actually wasn't the oil shock. This is a graph showing the price of oil and the inflation rate in the UK. And it's actually the inflation rate in the UK that you notice. So the oil shock created a spike in inflation, and you might think, well, you're only 11 years old, why would you notice that? When the price of a bar of chocolate goes up and the price of the bus tickets goes up that you need to get to school with, you do kind of notice these things. So that was a very, that was an interesting thing that happened, which really got me interested in solar energy. And the thing that's really interesting and it's obvious when you think about fossil fuels is they're a limited resource. This isn't rocket science. It's like if you have a jug of water, and you drink all the water, there's none left. So even from very early stage, I realized that solar energy was the most obvious, and I'll explain why, answer to the problems that we have with our current energy technologies. I'm not the first person, or sorry, let me explain why is that interesting. So the problem with fossil fuels is you basically just don't have enough. You can argue about how long they're going to last. Sure, absolutely. They're not going to last for 1,000 years. They're not going to last for 100 years. They're simply not going to last. This energy source, which currently powers the planet and powered it long before we were here, is going to last a lot longer than probably our civilization is going to last. And why is it useful? And really it's to do with numbers. So the amount of energy we use a year, the human race, is about 3.5 times 10 to the 5 petajoules. That's a big number. It doesn't really matter. Could have used it in different units, doesn't matter. The energy from the sun in 1 hour striking the planet is greater. So it's kind of a no-brain idea to not think about solar energy as a serious thing to study. I'm not the first person to realize it. Obviously, lots of people have. Siamesian in science in 1912, a very fine Italian photochemist, said, and this is wonderful, he said, if our black and nervous civilization based on coal and oil should be followed by a quieter one based on solar energy, that will not be harmful to progress or human happiness, which is a rather disturbingly prophetic comment, considering it was made over 100 years ago. And also Thomas Edison, in conversation with Henry Ford in 1931, said, and this is kind of a sad statement. I put my money on the sun and solar energy. What a source of power. And this is the sad bit. I hope we don't have to wait until oil and coal run out before we tackle that. And to be fair, people have tackled solar energy. It's just not so easy. It's not an easy nut to crack. Again, just pointing out the importance of it. There are various people. I've given a couple of talks to general audience. It's always interesting when you talk about energy sources because everyone has a favorite one. They either think biomass is going to work or hydro is going to work or geothermal or whatever. The problem is if humanity wants to live in the sort of way that we live right now, in 2050, we need to find 14 terawatts of energy, an additional 14 terawatts. And basically, you can't really do that from biomass or hydro. Geothermal, which is a sort of New Zealand favorite, is also, it's actually, technically, there's not 9 terawatts feasible. And wind and tide are a bit of a joke, especially when you compare them to this number here. So all of these are 1, 2, 10 terawatts. This is 36,000 terawatts on land, not on all of the world's surface, but on land. So it's clearly, this is a good option, I think. The problem, and the real crux of the matter is that to understand solar cells, you have to understand something called an excited state. You can call them different things. Physicists like to call them excitons. It doesn't really matter. I'll explain what they are in a minute. But basically, the nub of it is you have to understand something that's a bit tricky. So after I did my degree, as Richard said, I went to Los Alamos. And this is the sort of experiment you need to set up to understand excited states. So you've got a laser you can't see in this picture, another laser here, yet another laser here, yet a delay line, actually kind of a pseudo frequency doubling and continuum generation system, and a whole bunch of optics down here. You need exquisite alignment to actually measure these things. They're hard to measure. There's a reason the problem isn't solved. It's really, really hard to understand these types of solar energy type systems. The technicality of understanding them is very, very challenging. And so when I came here to Otago, I was really interested in these issues, the spectroscopy of electronic materials. And that included solar cells, conducting polymers, photocatalysts, and OLEDs. And when I was making the talk, I dug out this little OLED we made in the lab about 15 years ago now. And they're really great things to do. They're great things to study, but they're extremely difficult to study. So one thing that you can use is you can use all the innovations that you've got, all those experimental innovations that you try and get from studying something that's tricky to study, to actually maybe study, maybe use them in something that at least the sample isn't going to be tricky. And so that's what we've tried to do. We've tried to use our innovations in spectroscopy, which we've got from what I call molecular electronics, that side of our research, and apply it to analytical spectroscopy. And that's not to say one is more difficult than the other. They just have different challenges. But kind of using all that innovation over here to try and drive innovation in measurement at least, over in analytical spectroscopy. And so what can you measure? Well, this is some of the things we've measured: microplastics, cheese, milk powder, nectar, fruit and veg, bone, teeth, snake teeth, which are different from mammalian teeth, various other things. My personal favorite is rocks or geological samples, as I think the geologists we work with prefer to call them, and dried pig feces. We've actually measured dried pig feces, and I guess the joke in my research group is we'll literally try to measure anything if we think we can get something useful out of it. I think we can help people understand their analytical problem. What is spectroscopy? So as Richard alluded to, that's what I basically have done for my entire career. And I'll try and explain it this way. So it tells you about the types of molecules you have and their concentration in a particular place. How does it work? Well, it's a bit like if you think of a molecule like a tuning fork. If I bang a tuning fork, I get a note out, I get a particular frequency in the harmonics. Molecules are like multi-dimensional tuning forks. You can think of them as the chord that you might play on a piano, or this is actually what it looks like. There's a lot more notes, but basically the idea is that certain frequencies or energies give signals. The signals are either very high, or they're very strong, or they're much weaker. And from that, the bunch of squiggly lines there, or sharp lines, however you want to think of them, you can gain information about the material that you're looking at and you're trying to understand. So the bands are associated with specific vibrations, so you can provide information about molecular structure. We use a thing called Raman spectroscopy. It doesn't really matter what it is, it's just that it gives a unique signal. And basically it's like this. So if we think about, this is a very simple molecule, formaldehyde, and you can think of, there's a feature here which is due to the molecule kind of doing this, And then at the higher frequencies, the bonds are squeezing together. We're doing this sort of calisthenics for molecules, right? Exercise for molecules, and that's basically that pattern. We're able to work out what the molecule is and how much of it is present. So what about excited states? Why are they so hard? So a couple of wee diagrams. These are really nice. Super OLED technology and solar cell technology is just fantastic in places like Korea and China and Japan. These are world leaders in this area. And this just shows, these are actually solar cells that have been made into nice glass windows just to look pretty, because part of the problem is trying to sell them and get people to buy them. And then this one's quite nice. This is actually Los Angeles at night. It's kind of hard to see because the screen, which is made out of a plastic, is bent around. You see how it's basically a little scroll, like a piece of cellophane. For those of you who are a little bit older will know what I mean by that. But it's a piece of plastic you can bend around with a high resolution signal on it, and it's also can be made transparent. So excited states basically involve moving electrons. Molecules are made up of atoms and electrons, and the idea is that you move electrons from one place to another, and we call this the donor, which is where the electron came from. It's this orange circle or red circle, and it goes to the green box, which we call the acceptor. I'm simplifying hundreds of years' worth of work, but that's okay. You get the general idea that we made something different because we moved electrons around, and electrons hold molecules together. So that's one way of thinking about it. Let me give you a slightly more human example of that. So this is my mentor, my PhD supervisor, passed away a couple of years ago, very sadly, John McGarvey, that Richard did allude to. And John was not just a great scientist, he was a really, really good teacher. And not in the sense that he was theatrical, that's not what I mean. He had wonderfully insightful comments to make about chemistry. And one of the first ones he made, which really stuck with me, is to say, excited states are just not like the molecule they came from. And let me try and use this cartoon as an analogy of that. So if we have Stickman here on a seesaw, and I impart some energy with a photon, and Stickman was ready for it, so he flies up into the air and he goes into a little tuck, because that seems as sensible a thing as you can do in that situation. But he clearly has a very different shape up here than he had when he started out, and he clearly has quite a lot more energy. And he's also in a dynamic state. Yeah, he's not going to stay there. He's going to continue to move. And if you were able to ask Stickman, how are you feeling, he'd probably feel very differently than he did standing confidently on the seesaw. So he continues to move, and then he eventually plants the landing over here, and he's all good again. And that really says a lot about excited states. There are a couple of things to realize. They're not the same as the ground state. They're in a very different shape. They feel very different. If a molecule could feel, you understand what I'm saying. But the excited states are hard to measure because they're very short-lived, and they're also very reactive. One of the reasons we like to make excited states is because they can do reactions that ground states can't do. And the key things then are energy, lifetime, and reactivity. Those are the key things that one worries about in these situations. So let me give you an example. I'm not going to have too much chemistry here, but this is just a really wonderful example of much of the work we've done is to try and prove to people that looking at the structure of a ground state molecule doesn't really help. It's like asking Stickman how he feels when he's on the seesaw as opposed to when he's up in the air upside down. Yep. And so let's take a couple of molecules, and we look at these molecules, and they're all going to make excited states, and they're going to have a little dot plus over here. There's going to be a donor, a hole over here, and an electron over there. And you can see, look, I mean, this is nice and close. They're close together. Then I can pull them apart, and I can pull them apart, and I can pull them apart, and I can pull them apart. Now, if I ask the question, I have a hole, that's where the electron wants to be, and I've an extra electron over here, and I pull them apart, you'd quite reasonably say, well, obviously, it's going to take longer for this guy to get back than the two that are intimately connected. Yes, that seems reasonable. And indeed, charge separation theory would say that. You'd say, oh, you separate the charge, we're all good, going to make a longer excited state. Let's see what happens. So for the guys who are bound up together, they have a lifetime here. That means they're up in the air for 6,000 nanoseconds, 6 microseconds, right? The ones who are very close together. As we go further and further apart, the lifetimes drop to 4 to 2 to 1 microsecond. In other words, as we pull these apart, they're collapsing back in more quickly. Not less quickly, more quickly. Yeah? So there's a lovely, I mean, Isaac Asimov's Foundation, I think, has just been made into a series, but I remember reading the books when I was a kid, and Isaac Asimov had, I think, the most wonderful science quote ever. It's the best science quote, and it's this: the most exciting phrase to hear in science, the one that heralds a new discoveries, is not eureka, but Oh, that's funny. And this is a, oh, that's funny. Actually, sometimes it's not said with quite such a calm way. It's, oh, no. What's going on? What's happening, man? I mean, we've got the ones that are close together. It's a long lifetime. This one, we tried to spread it out. It's a short lifetime. What's going wrong? But if you analyze it and you think about it, and stop thinking about the molecule as it looks in the ground state and think, well, what does the excited state see? The excited state sees energetics. That's all it sees. You can perfectly adequately explain this using, I mean, sorry to be a bit technical here, but it's called Marcus-Hush theory. And it basically allows you to correlate that process. So it doesn't matter that it's further away. The excited state didn't care. It turns out the energetics driving it were less, and in the inverted region, that actually makes it easier for it to collapse back in. Seems a bit weird, but quantum mechanics can be a bit like that. It's just the way it is. So it gets worse. So this is another example, a really nice molecule that we actually, I shouldn't say, I didn't make any of the molecules. My colleagues did. I'll talk about them later on. We had this lovely molecule. It could give up, make a positive hole there and put an electron there, one charge transfer state, or it could put a hole there and put the electron over there, another. So it had two charge transfer states together, one called an MLCT, one blue and one red. Let's not worry about the labels. And if I put it in one solvent, I got one excited state, and then I put it in a different solvent and got a different excited state. And it turns out that if you do, and we did some pretty nice calculations on this, quite sophisticated calculations, as you structurally change the molecule, these subtle structural changes, the blue state goes down and then back up in energy, the red state kind of moves along and then goes down in energy. In other words, predictability is not something that you have a lot of. You need to understand the excited state, not what it looked like before you made it excited. So asking Stickman at the base of the seesaw ain't going to cut it. And so what I hope I've shown there, and I appreciate it's very, very rushed, and I've left a lot of stuff out, but excited states are unpredictable. They're hard to get data on. That's one of the reasons why we don't have as extensive predictive toolbox. But you cannot engineer solar cell materials or photocatalysts without understanding the excited state. And that's something that we've really indicated in the last couple of decades. So by characterizing excited states in systematic studies and comparing the experimental data and the quantum calculations, we've highlighted some of the real issues around that, and it's now kind of gratifying that people are really starting to focus on the excited states as a way of understanding what's going to go on. So harnessing the experiments in excited states to look at analytical problems. What do we do there? Well, there's a whole bunch of examples, and full disclosure, I'm a vegetarian, but here we have we've done a lot of studies with the meat industry, and one of them is that you can take spectra of lamb, venison, and beef. Those of you who are aficionados will know that they're different colors. Yes, I appreciate that. But they're not different colors while they're speeding along a conveyor belt in what I would call a slaughterhouse, but you call it a freezing works. So they're not so easy to actually distinguish those colors, but we can use the differences in these squiggly lines, right? They look very similar, but they're not the same. And using multivariate analysis, you can actually separate them out and therefore differentiate them in a reasonably rapid way. Another good example is krill oil. It's a great material to take. Fish oils are generally good for us, but krill oil is very nice because it has this astaxanthin in there. It's an antioxidant. It's actually physically a red color, which is why krill oil is the color it is. And it has this very distinctive spectrum, so it has these very two strong features. And when astaxanthin has, and there's krill oil, and you can see tuna oil, which doesn't have any astaxanthin in it, doesn't have those features. And again, you can build calibration curves whereby people can measure the quality of, not the quality, I apologize, the quantity of astaxanthin in the krill oil tablet without splitting the tablet open. You can simply focus your way through it. So that's great, but that's nice. We use some of our technology, some of the ideas we got from excited state to do some nice analytical measurements. It's always fun to push it a wee bit further, right? So one of the ideas that we started to come up with is we looked at the problems that analytical chemistry had, and one of them is around crystalline states. And so we started to look at what's called low-frequency Raman spectroscopy. So it's not obvious to you if you're not in the field, but these numbers down here are very small. So these are very low-energy vibrations. So earlier I said it's like calisthenics, right? For the molecule. The molecule's doing this, or it's doing this, right? That's one type of it's within the molecule. It's vibration within the molecule. Another way molecules can move is you imagine lots of me in a row. It's not a very nice thought, particularly with this guy. I know I'm very, very colorful. But if you imagine them just swaying backwards and forwards. So obviously, depending how I'm organized, that swaying backwards and forwards is going to be different, and that's what we call low-frequency Raman spectroscopy. The modes that are associated with it, you kind of showed it here, this is benzoic acid. And I guess the point I want to make is this is a little group we change from this species to this species to this species to this species. Look how different the squiggly lines are. The squiggly lines are very, very different, but all I did was make a wee change over here, and they all bound like this. So what I'm trying to say is that very small change made a big difference to how the molecules did this, right? Like I put a backpack on, and suddenly I was doing this in a very different way from when I take the backpack off, kind of like that. And those modes are often called phonon modes, but it doesn't really matter. They tell you about crystallinity. They tell you about the crystalline state. And we had a lovely colleague, Ben Boyd, who came over. I gave a talk at a conference. He gave a talk at a conference. I went up and talked to him about what he was trying to do. What he was trying to do is he was trying to understand a thing called ferroquin, which is a drug, and it was being digested in milk, and he was trying to understand it. So ferroquin is a crystal. So what you can do is you can take your milk sample, you can stick it in a small angle X-ray machine, and X-rays diffract crystals. They don't diffract things that aren't crystals, so that's all good. You can work it out that way. And I said, well, we can kind of do a similar thing, and indeed, he was able to do it in our lab with this experiment here. So there's a spectrometer, there are the various filters we use, and the various optics. So, I mean, all up, I don't know, say 60 grand maybe. And we bought nice stuff, because, you know, in New Zealand, it's important to buy nice stuff, because you may have to use it for a very long time. What we found was that this is what happened with his ferroquin. So his ferroquin dissolved away in about 10 minutes. And this is the experiment he did using small angle X-ray. The difference is, this is our experiment. This is the Australian synchrotron where he had to do his experiment. Now, I appreciate you can do a lot more in a synchrotron, but you ain't gonna take small angle X-ray and turn it into a handheld device. This stuff here, you can turn into a handheld device. There's a big, big difference technology-wise. Again, just making that point again, the crystalline state, so that business with us shaking together like we're in a concert or something like that, that turns out to give much, much bigger signals than when we're doing our calisthenics, right? So our calisthenics are right here, our moving together is here. And indeed, if I lose crystallinity, if I get all confused, instead of us being on a nice row doing this, we all get confused, then we end up with this sort of little hill, which you can also model. You can also use that information. So what do we try and do? We tried to do a bunch of stuff, because when you've got something new to play with, it's always cool to see what it can do. So the dissolution I've already talked about. We can characterize these different states. We can characterize materials in what are called solid dosage forms. They're actually the tablets that you're going to use, and I'll talk a wee bit about that just to finish up. And we can also do computational analysis and discovered some nice things about that. So one of the things, of course, you're trying to do something. So I'm originally a time-resolved spectroscopist, right? I'm interested in excited state, so I want to see how fast can we go, right? It's not an unreasonable thing to ask. It's a good question to ask. And this is a study we did where we dehydrated something. So this is peroxicam monohydrate in the anhydrous form. The key thing here is the structures are different. And as far as the law is concerned, this, let me get my arrow back. This is not the same as this. They're actually patented as different materials. So if you have a tablet with peroxide monohydrate in it, and it dehydrates while on the shelf to the anhydrous form, you've gone from being I'm licensed and legal to I'm not necessarily licensed and illegal. And the pharmaceutical companies spend a lot of time protecting, and not unfairly protecting, whatever they've got a license on or whatever they've got a patent on. What we found was we could measure this incredibly quickly in a few seconds. So we could actually, this isn't excited states anymore, this is just something happening fast, so we can measure it in a few seconds, which is really good. We're getting close, not quite at video rate, but we're getting there, we're getting there. And all of this data tells you about the nature of the molecules. And what happens in this case is the little water molecules loosen up, slip away, and then this reorganization occurs, and that's what we were able to show. So the last thing I wanted to do was, okay, so the other problem we have is what we'd really like to be able to do, we're talking about tablets. If you think about what a tablet looks like, so this is a little cartoon of a tablet, and it's blister pack. There's the tablet, and it's blister pack. It's actually made up of, that's the bit that's going to make you better, or hopefully make you better, the active pharmaceutical ingredient. It's right in the middle. It's got a big coating on it. It's got a big plastic covering on it for the blister pack. What you'd really like to do is to be able to see inside there and figure out what's going on without ripping open the blister pack or snapping open the tablet or any of that nonsense. And you can do that. You can do it using the craziest idea ever, which is called spatially offset Raman. And that is where I said earlier when we did spectroscopy, you always want to have things aligned. Never tell a laser spectroscopist that I've misaligned the system. It's very, very upsetting. So everything needs to be aligned. It actually turns out in this case, if I line everything up perfectly, I'm going to measure the blister pack. I don't care about the plastic. It's this I want to care about. It turns out as I start to move, as I start to differentiate between where the laser comes in and where I look, I make that bigger and bigger. I actually look deeper and deeper inside the sample. And it's called spatially offset Raman spectroscopy. It's actually used to work out what's in the liquids inside containers. We kind of put a twist on it. We used, well, hold on, our low-frequency Raman's really good at figuring out this crystallization. Why don't we put them together and we make what we call sulfurs, S-O-L-F-R-S. And there's two different ways of doing it. You can move the laser away that way, or you can defocus the laser this way. So does it work? Yep, it does. So basically what you can do, it's a really simple experiment, but perverse to a laser spectroscopist, is that you focus to maximize your signal. That's the top excipient. That's the one that's in green there. That's a polymer, so it's just got this smooth surface. And then as I defocus away, it's kind of perverse, we start to see the surface below, which is a lactose in this case. And then if I focus even further away, I actually get the bottom, the layer at the very, very bottom. So we're actually trying to see what we can do with that. It's kind of an exciting result that we've currently got. So I think I've tried to show that it's possible to measure complex materials and interrogate composition with high accuracy. It's possible to use low frequencies to measure rapid changes in subsurface structure and layering. And a lot of those innovations flow from excited state measurements, or actually possibly, I've simplified it, it actually flows from the fact that when you do excited state spectroscopy, you have to innovate. And if you start to do that with analytical measurements, you kind of get the same sort of good results. I just want to finish up. So this is Alan McDermott, who a number of you, I think, hopefully know, who got the Nobel Prize in Chemistry in 2000. He was from New Zealand, of course, and the McDermott Institute, of which I'm part, was named after him. And Alan had a couple of great sayings. So one of them is very famous, and you'll all, may not all know it, but it's well known. The first one that I'm going to give you is not so well known. Alan said that science without funding is fantasy. He didn't always use the word fantasy, but he basically said, nah, you ain't got nothing without some funding. You need to have funding to do these things. And so I think it's really important to recognize the people who funded us over the past couple of decades, the Dodd Walls Center, McDermott Institute and Redett Institute in particular, but also all of these other guys and many different types of different funding sources, and of course the university as well, which has also provided us with really good funding. The other thing that Alan said is science is people. He said this, of course, but he kind of appropriated it, I think is the right term. Because, of course, a much older saying is he aha ti mea neo te aho he tangata he tangata he tangata. What's the most important thing? It's people, it is people, it is people. So in the very last minute or so, I just want to thank these are just a list of the faculty and various researchers I've worked with over the years. And it's really been a privilege and a pleasure to work with them, but the people I really want to thank are these guys here. So I don't have photos of all the PhD students, but I got a fair few of them, right from the very earliest students back in the group in the early 90s, right through to the most recent one, Fatima, just down here. I just want a special call out as well to Sarah Miller, Dr. Sarah Miller, who's run my group basically while I've been HOD, and she's been fantastic in holding things together. But a lot of what I do is, I mean, it's nice to solve problems, but it's also nice to work with people who are really enthusiastic, and it's made a huge difference to being able to do what we've been able to do. And someone said, you're the smartest person in the room. I definitely don't think that. I've had the privilege of working with a lot of smart, dedicated people who've been really, really effective. We've been effective in working together. So last thing, I just want to thank these guys here. This is obviously not this guy here. You can already see him. But I want to just thank my family, my daughter Emily, son Callum, and most particularly my wife Catherine, who I realized I met in 1984. So she's been basically with me the entire time I've been a serious or even semi-serious scientist. So thank you very much for your time. Thanks.
Kia ora, Keith. Thank you for that wonderful expose through spectroscopy materials and other things. I am reminded also in your lecture of another famous New Zealand scientist who would have said, at times, if we don't have the money, we have to think. And so that ability to be able to think about an issue and find and get good ways to resolve it. You've also brought to us that wonderful legacy through Alan McDermott. I know the work that he performed as one of the discoverers of conducting polymers, plastics that conduct electricity, certainly started from an observation that was, that's funny or that's interesting, seeing something out of a plastic synthesis experiment that looked silver and gunky rather than clear and plasticky, that innate curiosity we shouldn't lose, and it's very clear from your work and the work you've built on that leads to both important discipline discoveries as well as principles that can be applied to the real world, up to and including dried pig feces. So can you all please join me again in thanking Professor Gordon for his lecture.
So our second Carl Smith Award medal today is for John Shaver, and I've already mentioned many dimensions of his work. So I'll just introduce him by giving you some details of his career. So he studied as an undergraduate at Indiana University of Pennsylvania in anthropology, and then went on to the University of Connecticut, UConn, where he took his MA and his PhD, completing it also in 2012. His first academic appointment following that was as a postdoctoral fellow at the Laboratory for Experimental Research on Religion at Masaryk University in the Czech Republic from August 2013. And this place is widely recognized as one of the leading institutes for the scientific study, the kind of scientific study of religion that John does. He came to New Zealand in the following year as a postdoctoral research and teaching fellow on a Marsden-funded project led by Joseph Bobulia at Victoria University in connection with the New Zealand Attitudes and Values Study. And from there, he then came to Otago in 2016, and here he has had a remarkable record of success in attracting grant funding, which has made a real difference to our program and to the division, I think. For his talk today, as we were discussing how he might squeeze into a few minutes the wide range of subjects and areas, sites that he's worked on, I encouraged him not to talk about that, but to present simply his boldest and most consequential idea. So if you don't like his talk, it's probably my fault. So John, will you please come now and address us on the topic of the promise of a scientific social science.
Tēnā koutou katoa, ko John Shaver tōku ingoa. E ahurangi tūhono o tikanga whakapono aho, no Pittsburgh aho, kei te mana whenua e mihi ana. Kei ngā rangatira o te whare wananga o Otako e mihi ana ano. No reira tēnā koutou katoa. I would like to begin by thanking my family. I think they're out there for their many years of support, and my colleagues in religious studies, the university more broadly, and internationally for their continued collaboration and encouragement. I'm especially appreciative of my mentors, Rich Joseph Bobulia, and Dimitris Zigalatis. But I would like to dedicate my talk and award to the anthropologist Roy Andrade, one of my former teachers and graduate supervisors. When I came to Otago in 2016, I emailed Roy to let him know that after 3 years as a postdoc, I'd finally secured a permanent position. I never heard back from him, and Roy passed away a few months later. I like to think that he at least read that email and that it made him proud. When I met Roy, he was deeply concerned with the future relevance of anthropology specifically, but also the general functioning of the social sciences more broadly. It seems strange to use this example in front of a physicist and a chemist, but the example of cold fusion illustrates one of the weaknesses of the contemporary social sciences. About 30 years ago, a flurry of scientific activity began when a group of chemists reported that they could fuse hydrogen atoms without producing heat. This report, however, contradicted what physicists knew to be the nature and structure of atoms. In short, the controversy boiled down to this: either the chemists were wrong or the physicists were wrong. In the natural sciences, the pursuit of knowledge about the world is a single enterprise, so teams of chemists and physicists got working to see who was right. It turns out, of course, and to the delight of a much younger Professor Blakey, that the physicists were right. Roy himself was trained in a way that embodied this ideal. He was trained at the Harvard Department of Social Relations, a department founded with the explicit goal of developing a comprehensive theory of human behavior, and one that focused on the interdisciplinary training of its students. Social Relations was founded by a group who refer to themselves as the conspirators, a group of scholars who formed a new department due to dissatisfaction with their traditional academic departments. They included prominent figures in sociology, namely Talcott Parsons, in psychology, Gordon Allport, and in anthropology, Clyde and Florence Kluckhohn, and Beatrice and John Whiting. It is noteworthy that many of these figures themselves had been trained in the humanities and medical sciences. Social Relations existed for about 25 years and produced some of the most noteworthy social scientists of the 20th century, including Robert Bella, Michel Rosato, Stanley Milgram, Clifford Geertz, and Roy Andrade, among many others. According to Roy, and of no surprise to any academic, the Department of Social Relations eventually disbanded due to internal fighting along traditional academic fault lines. Now, what does any of this have to do with my research? Well, Roy's intellectual commitments have had a strong influence on me and how I approach the topic of most of my research, religion. Rituals and supernatural beliefs exist in all known human societies. They stretch deep into the archaeological record, and across cultures, religions are structurally quite similar. The deep history, universality, and shared structure of religion suggest that religion is an important part of our evolutionary history, and that its study warrants evolutionary analysis. And indeed, one of the benefits of contemporary evolutionary theory is that it provides a unified theory of human behavior while simultaneously appreciating unique cultural variation. A theoretical framework much like the one the conspirators in social relations were hoping to develop. But religion is also extremely complicated, intricately intertwined with many aspects of human nature and culture, and as such, understanding its various components, their cognitive underpinnings, and the relation to society requires employing the methods typically associated with many disciplines. My research has used evolutionary theory to attempt to understand the origins, functions, and future of religion by employing biological, demographic, ethnographic, and experimental methods across diverse cultures. Today, I will touch on a few aspects of this research. In terms of ritual behavior, in experiments led by Martin Long and published in Current Biology, we showed that ritualized behavior spontaneously emerges under conditions of high anxiety. Using motion capture technology, we found that people who are stressed engage in more repetitive and rigid behaviors. The plot on the screen compares the hand movements of a stressed participant on the left with one in the low anxiety condition on the right. Visible clusters in A are indicative of recurring behavioral movement patterns. Now, rituals vary tremendously across cultures, but at their core, they all involve repetitive, redundant, and rigid behaviors. And this work demonstrated that these behaviors, that is, ritualized behaviors, spontaneously emerge at the individual level. But rituals occur within groups, and humans are not the only animals who engage in ritual. Indeed, at times there are striking similarities between non-human and human rituals. For example, young men on Pentecost Island in Vanuatu build towers for ritual performance, climb the 25-meter towers, tie vine to their feet, and jump while the community sings and dances below. That's the image on the left. Similarly, male bowerbirds annually build tall and highly decorated towers for ritual singing and dancing, the image in the middle. And much like people singing together on a church on Sunday, humpback whales periodically join together to engage in ritualistic song. Now, you may object that these are superficial comparisons, but at a fundamental level, ritual behavior across all those species who practice it is all about communication, not communicating to a god or gods, but to other group members. Since it is non-essential and not immediately directed toward survival or reproduction, how could ritual possibly be a product of natural selection? Developed in evolutionary biology, signaling theory assumes that individuals engage in the costs associated with ritual behavior, which are often substantial, to communicate important attributes about themselves to fellow group members. Even though they are costly, these behaviors can evolve because individuals materially benefit from the efficient and reliable communication that they provide. Prior to my work in the area, a dominant approach in evolutionary studies of religion had been to approach religious culture as a suite of adaptations for cooperation among unrelated individuals. Indeed, a significant body of empirical data indicate that religious people tend to be more cooperative with one another than their secular counterparts. However, this body of work downplayed the fact that cooperation doesn't always result in a 50-50 division of resources, and that religions often function as a means for those in power to exploit those without it. In Fiji, based on several years of long-term qualitative and quantitative fieldwork in a village in the rural north, I systematically investigated entrenched forms of social inequality legitimized by religious culture, and in particular from mission Christianity. On the screen in the distance, you can see the village where I used to live and conduct research. My fieldwork led to a series of publications revealing a darker side to religiously motivated cooperation. My work suggests that early Christian encounters came to reinforce and legitimize traditional chiefly authority, augmenting processes of social stratification and inequality that were present in the traditional caste-based social system, and today manifest as variation in contemporary ritual practice. This research helped to clarify why inequality persists and how elites manipulate costly ritual participation to conserve power in what is still a highly cooperative and largely redistributive system. In subsequent work with colleagues, conducted with Tamil Hindus in Mauritius, which is off the coast of Madagascar, we employed field experimental methods to quantify the costs of an extremely costly ritual, the kavadi, and extended the inequality model developed in Fiji. On the slide, you can see that participation in the kavadi includes several piercings and the carrying of large, heavy structures. The ritual also entails walking like this for long distances, about 10K in the hot tropical sun. Our study showed that participants' relative position in the social hierarchy systematically affected their form of ritual practice. Specifically, we found that low-status participants were more likely to engage in participation that required higher physical costs in terms of the number of body piercings as well as physical effort, while high-status individuals were more likely to use financial capital to present more elaborate and eye-catching signals. This work helped to clarify recurring features of religion and social inequality amongst distinct cultures, enabling generalization about key features of human behavior and motivation. Interestingly, in another study in the same location, but with data from a different kavadi ritual, our longitudinal study found that participation in this ritual, although incredibly costly, was associated with significant increases in mental health and well-being over time, but with negligible effects on physiological function. Here you can see a comparison of a group who participated in a control group who simply walked in the ritual procession without receiving piercings or carrying the kavadi. The circle on the graph on the left illustrates increases in mental health from baseline among people who participated versus those who were in the control group. In the circle on the right, you can see that people who experienced more pain reported greater increases in well-being from baseline. Colleagues and I speculate that the sensory, physiological, and emotional hyperarousal involved in strenuous rituals such as the kavadi can produce feelings of euphoria and the alleviation of pain via endocrine alterations in neurotransmitters such as endorphins or endocannabinoids, and buffer stress-induced systems on the immune system. In other words, these rituals can be good for you. Combined with earlier work, these results suggest that low-status individuals may experience greater mental health benefits precisely because the social system encourages them to pay greater costs. Now, recently I've been focused on understanding how religion may impact reproductive success. Despite the centrality of reproductive success in evolutionary models, prior to my work with colleagues, the link between religion and fertility had not been investigated systematically from an evolutionary perspective. This is especially surprising because because across the world, religious people tend to have higher fertility. This fact is relatively well known across the social sciences, but from an evolutionary perspective, the high fertility of religious communities is puzzling. Humans face trade-offs in how to best allocate the limited resources they have available to them. One critical trade-off is between the quantity and quality of their offspring. The more children that parents have to care for, the less time and energy parents can invest in each child. And fewer investments in a child are associated with poor developmental outcomes. Studies have documented this trade-off and found that sibling number is negatively related to a child's cognitive and physiological development, as well as their socioeconomic success in adulthood. We would therefore expect, due to differences in family sizes, children born to religious parents would fare less well in terms of developmental outcomes than children born to secular parents. Yet, while there have been few studies that compare the success of religious and secular children, the available evidence suggests that children born to religious parents fare as well as those born to secular parents. What then can explain this paradox of religious fertility? We have attempted to integrate two lines of research to propose and test a solution to this paradox. First, and as mentioned earlier, studies consistently find that religious people tend to be more cooperative with one another than the members of secular groups. Second, alloparenting, or the provisioning of childcare by individuals other than the parents, has been shown to positively influence a woman's fertility and improve children's survival, health, and developmental outcomes. We hypothesize that religious cooperation extends to alloparenting, and that the higher levels of cooperation among religious mothers can help to explain their higher fertility. We also predicted that the support that religious mothers receive would positively impact child developmental outcomes. In other words, we propose the idea that religious cooperation mitigates the costs of high fertility to children. In a study with 12,980 New Zealanders enrolled in the New Zealand Attitudes and Values Study, we found that cultural groups with higher fertility, specifically Maori and Pacifica, were more likely to alloparent than cultural groups with lower fertility. We also found that religious New Zealanders, after adjusting for ethnicity, were more likely to alloparent than their secular counterparts. These findings suggest that fertility levels correspond to levels of alloparental support, and because religious New Zealanders have more of this support, they are able to achieve higher fertility. In a more recent article, my collaborators and I investigated this paradox further using a large longitudinal database of mothers and their children from Bristol, UK, known as the Children of the 90s Health Study. We first investigated whether women who invest in ritual behavior benefit from stronger support networks and increased cooperation among co-religionists. Over 10 years of data, we found that church attendance was positively associated with both a woman's social network support as well as the frequency with which they receive help from co-religionists. Religious women have more social resources at their disposal. Aid from co-religionists was positively related to a woman's fertility as expected. Women who receive more cooperation from members of their congregation have more children over time. In terms of developmental outcomes, on this plot, each colored point reflects a child's test score on standardized tests administered when the child entered school, and again when they were 7, and then when they were 8. Points to the right of the dotted line represent a positive association with test scores, and those to the left represent a negative association. The first thing to point out is that there's a negative relationship between sibling number and child test scores at each of the 3 time points we examined. Kids who have more siblings do worse on standardized tests. The second, more general thing to point out is that, and this is relatively well known, that these tests are biased. For example, a woman's education, a mother's education, and mother's household income were both positively associated with a child's test score at all 3 time points. The models further reveal a positive association between mother's social support and aid from co-religionists on a child's test score, particularly at later stages of development. This suggests that women's social networks positively affect child cognitive development, and that the greater levels of social support among religious women may help to explain why these women exhibit higher fertility without the same negative effects on children's development as secular women. These relationships may also help to explain the resilience of religion across the globe today. We are currently testing these hypotheses further with fieldwork in the US, the Gambia, Malawi, India, and Bangladesh. I don't know why, but I feel like I should quickly point out that I'm a childless atheist. To return to the analogy with cold fusion introduced earlier, our work on the evolution of religion and on religion's effects on social inequality, mental and physiological health, fertility, and child development are unlikely to cause other social scientists to rethink much of what they had previously thought about religion, or to check to make sure that we've got it right. At the end of his life, Roy Andrade was deeply discouraged by the fact that most theories in the social sciences have become intertwined with Western models of morality, having emerged from external politics, and are thus built upon explicit moral assumptions about the world. For example, many Western social scientists assume that hierarchies are inherently harmful and develop theories whose ultimate aim is to emancipate people from their oppression. But hierarchies are valued in many cultures, and empirical research suggests that hierarchical social organization can be more productive than egalitarian structures, and that the costs and benefits of social organization are different for groups of people than they are for individuals. But moral models don't readily permit counterfactual analysis, because they assume to know in advance what is good or bad about the world, rather than attempting, as objective as is possible, to simply figure out about the world prior to attempting to effect change. Another example that occurred to me last night was that after I presented the COVID research a couple years ago at the Otago Global Health Institute, someone came up to me afterwards and said, "Those images were pretty graphic. Don't you think you should have issued a trigger warning before you showed that to us?" And I thought maybe I should do that. And then I remembered that I saw that some research just examined the effectiveness of trigger warnings, and I looked at that, and that research suggests that trigger warnings either do not do anything, or amongst people who see trauma as central to their identity, they make things worse. So trigger warnings do nothing or cause greater PTSD in the very people they're meant to protect. Roy advocated that as social scientists, we should keep our models of our responsibilities as researchers separate from our models about the world, or else the result is, and I quote, "very bad science and very confused morality," end quote. Indeed, moral models have tended to prohibit interdisciplinarity and the progressive accumulation of knowledge, further entrenching disciplinary silos. In a review of their historical and cultural development, Roy recognized that while moral models had become commonplace across the social sciences, there were few scholars working with theories and methods that had the potential to encourage the interdisciplinarity that he had advocated throughout his career, but had seen all but disappear. Although he didn't employ evolutionary theory much in his own research, he recognized that contemporary work in the area had the potential to be a strength to researchers across many fields. Indeed, my own work has only been possible because of the collaboration of fellow anthropologists, as well as biologists, demographers, neuroscientists, psychologists, public health researchers, sociologists, statisticians, and religious studies scholars. I'd just like to conclude by thanking them and by thanking you, Roy. Tēnā koutou katoa.
Kia ora koutou, Ben, John, thank you very much for your very insightful lectures, and thanks to all three lecturers. I think what we've seen today, and actually in bringing the DRM and the Carl Smith lectures together, we've seen some of the values of being able to undertake research and teaching at a comprehensive university like Otago. That what you've shown us is that those principles that actually in New Zealand are embedded in the law, in our Education Act, of our requirement to preserve and enhance academic freedom, to challenge conventional wisdoms and be critical and conscious of society, are values that we hold dearly and that you have advanced in your scholarly work. I do look forward to the day, though, where we can see some of the crossovers into the hard sciences, and perhaps Professor Gordon can apply Raman spectroscopy to some of your manuscripts, and actually we can think about excited states in a religious sense, and lead that into Professor Gordon giving some better analogies for the human nature of his work. I am, though, reminded, and actually I will just take one moment to to think that the challenges we provide within our disciplines shouldn't be blood sports. We should be collegial around our interaction, and being critic and conscious of society does not mean we have to be antagonistic. We can be critical friends of each other's and the discipline and of the communities we serve. And I do recall hearing a Nobel laureate, Tony Leggett, talk about his work to school children. He received the Nobel Prize for theoretical work around understanding high-temperature superconductivity. But his training, albeit in the Oxford tradition, had come through a BA and followed by a PhD in theoretical physics. He said that one of the things that attracted him to theoretical physics was at the time the discipline was not like some of the other disciplines, that it was a blood sport. It was about status. But as a theoretical physicist, he could put forward concepts and theories that were able to be tested by experiment, that he could be wrong without being stupid. So I think we should encourage always that idea that we can put out bold ideas and be confident that they can be tested and challenged against others' ideas. And if we are wrong, then I think in Keith's word, that's interesting, that's funny. So can you please all join me in thanking all of our medal recipients? And we should move on to the fact that we have to actually present with you your medals. And so I will now do that and first invite Professor Gordon up to receive his Distinguished Research Medal. I think we have a photographer here. The other thing that's being done differently, this evening there is a university-wide awards ceremony for the research awards, teaching awards and general staff awards. Professor Gordon is not able to make it to the function, so he gets his certificate tonight as well. But I think both Ben and John are able to come, so you just get your medal, your book, and another little gift. So perhaps we'll get Ben to come next to receive his Carl Smith medal. Last but by no means least, John, please come up. Finally, but before we go, just to identify that ritual and tradition are important. We not only build on the traditions of our forebears, but we create and enhance new traditions. I will close with a karakia, a closing benediction, the translation of which is to the form of lighten the weight of this occasion. So let's not take ourselves too seriously, so that the spirit, heart and body can be light and free as we depart. Orongo above, let this all be done in unity. So I'll close with that. Unuhia, unuhia, unuhia ki te uru tapanui. Kia wātia, kia mama, kia nākau, kia tīnana, te wairua I te ara takata. Koia I rongo e whakairia ake te ringa, tīna huie tā hiki e. Thank you very much, and all the best for the rest of your days. Thank you.